explains the mechanism of ATP synthesis. ATP synthesis is linked to the development of the proton gradient across the membrane. The proton accumulates towards the inside of the thylakoid membrane in the lumen. Let's discuss the steps that help develop the proton gradient along the membrane. Water splitting takes place on the inner side of the membrane, producing protons or hydrogen ions that accumulate within the lumen of the thylakoid. The movement of electrons to photosystems enables the movement of protons across the membrane. This is because the primary electron acceptor located towards the outer side of the membrane transfers its electron to H carrier. Thus, a proton gets removed from the stroma while transporting an electron. When this molecule passes its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the membrane. The Atp reductase enzyme is located on the stroma side. The electrons that come from the acceptor of the electron of the PS1 and the protons are necessary for the reduction of NADP+. These protons are also removed from the stroma. Thus, protons in the stroma decrease in number, while in the lumen, there is an increase of protons. This creates a proton gradient across the thylakoid membrane. The breaking of the proton gradient releases energy. The Atpase enzyme present in the transmembrane channel helps in the breakdown of the proton gradient. The Atpase enzyme consists of two parts. The F0 part embedded in the membrane, forming a transmembrane channel that facilitates the diffusion of protons across the membrane. The F1 portion of ATP synthase protrudes on the outer surface of the thylakoid membrane to the side that faces the stroma. The breakdown of the gradient provides energy to cause conformation change in the F1 particle of the Atpase. This makes the Atpase enzyme synthesize several molecules of ATP. The synthesis of ATP thus requires a membrane, a proton pump, a proton gradient, and the enzyme Atpase. Next, the NADPH and ATP produced by the movement of electrons are used in the Calvin cycle. So far, you learned that light reaction results in the formation of ATP, NADPH, and oxygen molecules. Now let's see how plants utilize these molecules to prepare food through the Calvin cycle. Oxygen, released during photosynthesis, is released into the atmosphere through stomata present on the leaves. While the ATP and NADPH molecules are used in the synthesis of carbohydrate from carbon dioxide and water. This phase is called the biosynthetic phase, as it involves the production of food. It is also called dark reaction. Dark reaction takes place in the stroma of chloroplast.